There is a cave in the Altai Mountains of southern Siberia that sits at the intersection of three river valleys, carved into a limestone cliff above a cold, fast-moving river called the Anui. Local people have known about this cave for centuries. They call it Denisova, named, depending on which version of the story you believe, after an 18th century hermit named Denis, who supposedly lived there and raised bees in the shadows of its chambers. It is not a dramatic cave by the standards of caves that have made it into the scientific literature. It does not have the soaring cathedral chambers of Lascaux or the dramatic vertical shafts of Spain's Pit of Bones. It is modest, a few connected rooms carved by ancient water into pale limestone, with a natural chimney in the ceiling that lets in both light and rain. Bears have denned in it for tens of thousands of years. The sediment on its floor is a compressed archive of biological history: pollen, animal bones, charcoal from ancient fires, stone tools, and scattered fragments of bone so small and so degraded that for decades, archaeologists sifting through them could not tell whether they were looking at the remains of animals or humans. It was in one of those nondescript fingernail-sized fragments of bone that science found something it was entirely unprepared for. And what it found in that bone did not just add a new name to the human family tree, it dissolved the assumption that the human family tree was even a tree. It revealed that our ancestors were living in a world shared by other kinds of people we had never seen, never named, never imagined, and that those people left a mark on us so deep and so permanent that it is still shaping the bodies of living human beings today, on the highest mountain plateau on Earth, in the deepest jungles of Southeast Asia, on the tropical islands of the Pacific. The Denisovans are the greatest ghost in human history, and the story of how we found them, what they did to our understanding of human evolution, and how they continue to reveal themselves in the bodies of the living, is one of the most extraordinary scientific stories of the 21st century. The cave was being systematically excavated by Russian archaeologists from the Institute of Archaeology and Ethnography in Novosibirsk, led by Anatoly Derevianko and his colleagues, who had been working the site for decades. The sediment layers in Denisova Cave span more than 280,000 years of continuous occupation, not just by humans, but by bears, hyenas, woolly rhinos, cave lions, and a rotating cast of Ice Age fauna that used the shelter of these chambers in the Siberian cold. In 2008, during routine excavation of one particular sediment layer, a researcher lifted a fragment of bone that was about the size of the tip of a finger. It was a piece of a pinky finger, to be precise, a distal phalanx from what appeared to be a juvenile individual, small enough that identifying it as human rather than animal required careful examination. The bone was sent to the Max Planck Institute for Evolutionary Anthropology in Leipzig, Germany, where a team led by Svante Pääbo had been developing and refining techniques for extracting ancient DNA from degraded archaeological specimens. Pääbo was already the most accomplished ancient DNA researcher in the world. He had spent years developing the methods needed to recover genetic material from bones tens of thousands of years old, a process complicated at every step by the tendency of DNA molecules to break apart and degrade over time, by the near-universal contamination of ancient specimens with modern human DNA, and by the sheer improbability of finding intact genetic sequences in material that had been sitting in cave sediment for geological ages. He had recently sequenced significant portions of the Neanderthal genome, the first time genetic material had been successfully recovered from a species other than Homo sapiens from the deep Pleistocene, and the methods his team had developed for that project were exactly what was needed for the Denisova fingerbone. What happened next was so unexpected that Pääbo has described the moment of seeing the results as one of the most shocking moments of his scientific career. The mitochondrial DNA extracted from the Denisova fingerbone did not match Homo sapiens. It did not match Neanderthals. It matched nothing in any known database. The sequence diverged from modern human mitochondrial DNA at a distance roughly twice as great as the divergence between modern humans and Neanderthals. This was not a variant of a known species. This was not a regional population of Homo sapiens with unusual genetics. This was something else entirely, something that had last shared a common ancestor with the lineage leading to modern humans and Neanderthals. Approximately 1 million years ago, far deeper in evolutionary time than the Neanderthals split from our lineage, which occurred somewhere between 400 and 800,000 years ago. In 2010, Pääbo and his team published the results in the journal Nature. They announced the existence of a previously unknown group of archaic humans, named after the cave where the fingerbone was found. They called them the Denisovans, and the scientific world stopped. The word that gets used most often to describe what the Denisovans represented is unprecedented. And that word is exactly right, and not just in the obvious sense that a new human species had been found. What was truly unprecedented was how they had been found. In the entire history of paleoanthropology, which stretches back to the first recognition of Neanderthal bones in Germany in 1856, Every new species of archaic human had been identified the same way. You found bones. You described the bones. You named a species based on the bones. The bones came first. The genetics, when they eventually became available, came second. With the Denisovans, there were no describable bones. There was a fingertip. And the genetics was the entire species. Svante Pääbo later described it exactly this way. It was the first time in history that a new group of distinct humans was discovered entirely through genetic analysis rather than through anatomical description. The Denisovans exist as a named entity in the scientific literature, not because a skull was found and classified, but because a DNA sequence from a tiny bone fragment refused to match anything that science had ever seen before. They were conjured into existence by their own genome. But let us be precise about what that genome said, because the details are where the real shock lives. The full nuclear genome, the complete inherited DNA from both parents, as opposed to the more limited mitochondrial. Genome inherited only from the mother, was subsequently sequenced from the same fingerbone fragment using less than 10 milligrams of powdered bone. 10 milligrams is approximately the weight of a grain of rice. From that grain of rice's worth of material, scientists generated a genome sequence of higher quality than many genomes sequenced from living people today, with each position in the Denisovan genome covered, approximately 30 times over, reducing sequencing errors to a vanishing minimum. This genome revealed three things that rearranged the human evolutionary landscape completely. First, the Denisovans were a sister group to the Neanderthals, not to modern humans and not to Homo erectus, but to Neanderthals specifically. The two lineages had diverged from each other somewhere between 400,000 and 500,000 years ago, after both had already split from the lineage leading to modern Homo sapiens. This placed the Denisovans and Neanderthals as the two most closely related extinct relatives of living humans, twin branches growing from the same point on the family tree, one spreading west into Europe as the Neanderthals, the other spreading east across Asia as the Denisovans. Second, and here is where the floor drops out from under everything, the Denisovan genome was not just an ancient curiosity preserved in a Siberian cave. It was alive. Fragments of Denisovan DNA, preserved in the genomes of living people through ancient interbreeding, were found in modern human populations across a wide geographic range. People alive today carry Denisovan genes. Third, and this is the detail that the popular coverage of the discovery consistently undersells, the Denisovan genome showed traces of something even older. Hidden within the Denisovan DNA were sequences that did not match Neanderthals, did not match modern humans, and did not match any known archaic human group. Somewhere in the deep past, the ancestors of the Denisovans had interbred with a population that was genetically further from any known human lineage than Neanderthals are from us. A ghost within a ghost, an unknown species that interbred with the Denisovans before the Denisovans interbred with us. The human family tree at this point stops looking like a tree and starts looking like a river delta, branching, merging, dividing again, carrying genetic material across species boundaries in all directions simultaneously. Now, let us talk about the teeth, because the tooth is where this script's title lives, and it deserves its own space in this story. In the same cave, in the same sediment layers, alongside the finger bone, researchers found two enormous molars. Molar teeth so large that when they were first examined by physical anthropologists, the scientists assumed they must belong to a different species from the finger bone entirely. The teeth were massive, their crowns bearing a morphology that looked more like Homo habilis or early Homo erectus, much older human ancestors from Africa, than anything you would expect from a species living in Siberia 50,000 years ago. They did not look like Neanderthal teeth. They did not look like modern human teeth. They looked ancient, primitive, and outsized in a way that seemed inconsistent with a creature that was, genetically, our close cousin. DNA analysis confirmed that the teeth belonged to Denisovans, to different individuals from the population represented by the finger. Bone, but to the same lineage, meaning that the Denisovans were carrying very large, very primitive-looking teeth while simultaneously carrying a genome closely related to Neanderthals, and while interbreeding with populations of modern Homo sapiens. They were, physically, a mosaic of traits that did not fit into any single expected category. They are a species science cannot describe beyond their DNA, because the physical evidence consists of a fingertip, a couple of enormous teeth, and a jaw, and not one complete skull has ever been found. The tooth that was reported in 2015, a second molar extracted from the same Denisova cave sediment, this time pushed deeper into the timeline to approximately 100,000 years ago, making the Denisovan population present in that cave, significantly older than previously established, delivered another layer of revelation. The Denisovans had been in that cave, in those Siberian mountains, for at least 100,000 years before the youngest traces of their occupation disappear from the record. They were not a transient population that passed through once. They were persistent, territorial, present across enormous sweeps of time in the same location, occupying the same cave alongside Neanderthals during overlapping periods, and as we will see, doing things in that cave that no one was prepared to discover. In 2012, another fragment of bone was pulled from the Denisova cave sediment. It was a long bone fragment, roughly 2.5 centimeters long, catalogued among more than 2,000 other visually unidentifiable bone fragments waiting in the collections for analysis. It sat there for 4 years. In 2016, a team at Oxford University led by Tom Hyam applied a technique called zoo-MS, zoo archaeology by mass spectrometry, to the collection. Zoo-MS works by analyzing collagen protein fragments extracted from bone, comparing their amino acid sequences against reference databases to determine which animal a bone came from. It is fast, inexpensive, and can process hundreds of fragments in the time it would take to sequence the DNA of a single sample. Working through the 2,000 anonymous bone fragments from Denisova Cave, Hyam's team flagged this particular fragment as human. It was sent to Leipzig, the DNA was extracted, and the result that came back made Vivian Sloan, the lead researcher on the analysis, pause at her computer screen and read the data multiple times before she believed what she was seeing. The fragment was from a girl. She was at least 13 years old when she died. She had lived approximately 90,000 years ago. Her genome contained equal contributions from 2 parental lineages, and those 2 parental lineages were not both Denisovan. Her mother was a Neanderthal. Her father was a Denisovan. She was a first-generation hybrid, not a person carrying ancient traces of interbreeding from hundreds of generations back, as the modern human populations that carry Denisovan DNA do. A person whose actual parents, one living and breathing Neanderthal woman and one living and breathing Denisovan man, had met in or near this cave in the Altai Mountains 90,000 years ago and had a child together. The Neanderthal mother, the genome further revealed, came from a population more closely related to the Neanderthals who would later live in Europe than to the Neanderthals previously found in the Denisova cave itself, meaning she or her ancestors had migrated east across Eurasia, traveling from the west to these eastern mountains before this encounter took place. And the Denisovan father carried within his own genome traces of Neanderthal ancestry from 300 to 600 generations before his lifetime. Evidence that Neanderthals and Denisovans had been interbreeding periodically for hundreds of thousands of years before this specific child was born. She was given a nickname, Deni, a 13-year-old girl whose parents were members of 2 different human species, who was born in a Siberian cave 90,000 years ago, and whose bone fragment lay unnoticed in a collection of 2,000 anonymous pieces for 4 years before science finally understood what it was holding. Deni is the only confirmed first-generation hybrid between 2 archaic human species ever found anywhere in the fossil record. She is, in a sense, the most extraordinary individual human being, in the broadest possible meaning of the word human, ever discovered. What Deni's genome confirmed, in the most intimate and personal way possible, is something that the broader genetic evidence had been suggesting since the Denisovan genome was first sequenced. The archaic human world of the Pleistocene was not a world of isolated species separated by genetic incompatibility and mutual avoidance. It was a world of populations in contact, populations that recognized each other as potential mates, populations that produced viable offspring together across species lines that science had previously assumed were impermeable. Neanderthals and Denisovans had been interbreeding on and off for much of their shared history. Modern humans interbred with both. And the genetic traces of all of this mixing are still circulating in the human gene pool, still shaping the bodies and physiology of living people in ways that researchers are only beginning to fully document. The most dramatic example of this is the EPAS1 gene. This gene regulates the body's response to hypoxia, low oxygen levels, by controlling the production of red blood cells and hemoglobin when the oxygen concentration in the atmosphere drops. At high altitudes, the air contains less oxygen per breath, and most people who travel from sea level to extreme altitude suffer the consequences. Altitude sickness, in its severe forms, can kill. The body of someone not adapted to high altitude responds to hypoxia by dramatically increasing hemoglobin production, thickening the blood, raising the risk of blood clots, cardiovascular complications, and dangerous complications during pregnancy. For most people, extended life at altitudes above 4,000 meters is physiologically brutal. But the Tibetan people, the Sherpas, the yak herders, the nomadic communities of the Tibetan Plateau, have lived at these altitudes for generations without the severe physiological consequences. Their bodies have been adapted to the thin air. And when researchers sequenced the EPAS1 gene in Tibetan people and compared it against the same gene in populations from lower elevations, they found something that they describe in the scientific literature as remarkable. The Tibetan variant of EPAS1, the one that provides the altitude adaptation, is almost identical to the EPAS1 variant found in the Denisovan genome. It does not match any modern human population from outside the high altitude regions of Asia. It does not match Han Chinese populations, even though Han Chinese and Tibetans diverged from the same ancestral group only a few thousand years ago. It occurs at high frequency only in Tibetans and in 1 or 2% of Han Chinese, and it occurs in essentially no other human population on Earth. The conclusion is unambiguous. At some point after the ancestors of Tibetans and Han Chinese diverged from their shared ancestral population, the Tibetan lineage encountered Denisovans. They interbred. And among the genes that passed from the Denisovans into the ancestors of Tibetans was this variant of EPAS1, which had been refined by hundreds of thousands of years of Denisovan adaptation to high-altitude environments, and which provided an immediate, powerful survival advantage to any modern human who carried it and subsequently settled on the Tibetan plateau. The Denisovans had been living at high altitude, as confirmed by the 160,000-year-old Siahi mandible found in the Baishia karst cave on the Tibetan plateau, a site that sits at 3,180 meters above sea level, for long enough that their bodies had evolved a sophisticated genetic response to the oxygen-thin air. When modern humans arrived and interbred with them, they inherited this adaptation. And because it was so powerfully beneficial in that environment, natural selection spread it rapidly through the Tibetan population. Until today, most Tibetans carry the Denisovan version of the gene, while the rest of the world does not even know it exists. There is something almost unbearably poignant about this finding when you sit with it long enough. The Denisovans are gone. They left no civilization, no recorded language, no art that we have identified, no cultural inheritance that we can point to and say, this was theirs. They exist in the scientific literature as a handful of bone fragments from a limestone cave in Siberia, a jaw from a Buddhist holy cave in Tibet, a tooth from a cave in Laos, and a genome reconstructed from less material than a grain of rice. But they are not gone from the world. Every Sherpa who guides climbers up Everest without an oxygen tank at extreme altitudes carries a piece of Denisovan DNA in the EPAS1 gene that allows their body to do what most human bodies cannot. Every Melanesian person in Papua New Guinea whose genome is up to 5%, Denisovan carries it through it. Biological legacy from a species that walked the earth tens of thousands of years ago, and then, at some point we still cannot precisely determine, simply stopped being there. The Inuit populations of the Arctic, who carry Denisovan-derived genes that help regulate body fat and cold tolerance. The populations of the Philippines, whose Denisovan ancestry is genetically distinct from the ancestry carried by their nearest neighbors, suggesting a separate interbreeding event with a separate Denisovan population at a separate time and place. Modern human immune genes, genes that help recognize and fight pathogens that appear to have Denisovan origins and may have given early populations in Asia a survival advantage against diseases they encountered as they spread across new territories. The Denisovans did not disappear cleanly. They dissolved into us. Gradually, over tens of thousands of years, as modern human populations expanded across Asia, the Denisovan populations that encountered them gave up pieces of themselves, pieces of their genome shaped by hundreds of thousands of years of adaptation to some of the most challenging environments on Earth, and those pieces persisted because they were useful, because they made the people who carried them better at surviving at high altitude, better at tolerating cold, better at metabolizing the fats available in Arctic environments, better at fighting off diseases in tropical Southeast Asia. The ghost did not haunt us, it helped us. And yet we still know almost nothing about what they actually were. We have never found a complete Denisovan skull. We cannot describe their face. We cannot reconstruct their body from enough skeletal material to get a meaningful picture of their physical form. The teeth suggest they had large, primitive-looking jaws. The jawbone from Tibet suggests a robust mandible without a modern human chin. The genome suggests their skin was dark, their eyes were brown, and that they carried gene variants associated with features we cannot easily interpret from DNA alone. An attempt to reconstruct a Denisovan face from DNA-based predictions of skeletal anatomy was published in 2019, producing an image of a wide, flat face with prominent brow ridges and a large jawline. But the researchers themselves were careful to note that this was an inference, a best guess, not a portrait. We have never seen a Denisovan face. We may never see one. The fossil record for this species is so thin that even with all the excavation power of modern paleoanthropology directed toward finding them, the physical bones simply may not have survived in any place we have yet looked. What we have instead is their genome, distributed across the living human population, like a message written in a language that took us until the 21st century to learn to read. The Max Planck Institute's ancient DNA laboratory, where Pabo and his colleagues built the techniques that made all of this possible, was awarded the Nobel Prize for Physiology or Medicine in 2022, a recognition not just of the Denisovan discovery, but of the entire field of paleogenomics that the Denisova cave fingerbone helped to inaugurate. Pabo accepted the prize on behalf of a scientific revolution, the revolution that proved you do not need a skeleton to know a species existed, that DNA can be recovered from material far older and far more degraded than anyone previously thought possible, and that the human story is encoded not just in the fossils in the ground, but in the living bodies of everyone walking the earth today. There are right now teams of researchers working in caves across Asia, in China, in Laos, in the Philippines, and Indonesia, searching for more Denisovan material. They are sorting through bone collections using zooms to find the human fragments hiding among thousands of unidentified animal pieces. They are extracting DNA from cave sediment from the ancient soil itself, pulling mitochondrial sequences from dirt that has not been disturbed in 100,000 years. The Baixia Karst cave on the Tibetan Plateau has yielded Denisovan DNA from 4 separate sediment layers, the youngest of a. The picture that is emerging, slowly, from all of this work is of a species that was far more geographically and ecologically adaptable than the paucity of their fossil record initially suggested. Denisovans occupied the cold steppes of Siberia. They lived at extreme altitude on the Tibetan Plateau, where the AIR contains 30% less oxygen than at sea level and winter temperatures drop to 40 degrees below zero. They were present in the tropical forests of Southeast Asia, in Laos, possibly in Indonesia, possibly in the Philippines. They survived across this enormous geographic range, from the Arctic edge of Siberia to the equatorial belt of Southeast Asia, across hundreds of thousands of years of Pleistocene climate fluctuation, ice ages and interglacials, sea, level rises and falls, ecosystem transformations. They did this with a brain we cannot measure, with tools we cannot confidently attribute to them, with a culture we cannot reconstruct, and with a face we cannot see. They were everywhere, and they left almost nothing behind except the biological inheritance that lives in the bodies of millions of people today, and the smallest possible sample of bones and teeth in the cold mud of a limestone cave in Siberia. That cave sits above the Anhui River in the Altai Mountains, still being excavated, still surrendering fragments of the people who sheltered in it across 280,000 years of human time. The hermit Denis, for whom it is named, has been dead for 3 centuries. The Denisovans, who gave their genome to science, have been dead for at least 45,000 years, but in the bodies of the people who live on the Tibetan Plateau, in the highlands of Papua New Guinea, in the islands of the Pacific and Southeast Asia, in the Arctic communities whose ancestors survived the cold, the Denisovans are still here, not as a species, not as a memory, but as a biological gift pressed into the human genome at some forgotten moment of contact in an ice age landscape, and passed down generation after generation until the day a grain of rice's worth of powdered bone dust told us for the first time that they had ever existed at all.